The cell therapy field is really just one of, I think, the incredible triumphs of immunology and applying basic immunology to the clinic. I remember being in a seminar where Jim Allison, a Nobel Prize winner, presented some of the first patients who had been treated for melanoma with the drug that would become Yervoy. And you could just see the amazement of everyone in that room to see, the things that they had done in mice, that they had done in cell culture, really get applied to patients. And I think that was the first time that I really understood that this basic field that I was just scientifically interested in had the potential to really impact people's health. So to go from that time when we were, studying monoclonal antibodies and their ability to treat tumors, to now when we're pushing forward in the cell therapy field, it's like seeing science fiction come to life. It's taking this thing that seemed incredible and impossible and watching patients really be impacted by it. One of the most important mechanisms of action of CAR T drugs is that they can kill tumor cells directly in the patient. And so to measure the potency of a CAR T cell, it's really important to be able to measure their ability to kill a target cell in vitro. When you're developing a potency assay for CAR T cells, you have to make a choice about what functions you're going to measure. And originally, a lot of developers chose cytokine production because we have great tools for measuring cytokine production. But a more direct measure of the potency of a CAR T cell and its efficacy against a tumor is target cell killing. And that's why we developed the high-bit target cell killing platform. So this assay is based on a split luciferase technology where the detection reagent contains a larger component of the luciferase called large bit, and the target cells have been engineered to express a high-bit protein tag. So when the target cells are alive, that high-bit tag is sequestered inside and is inaccessible to the large bit in the detection reagent. But when a CAR T cell comes along and kills that target cell, it releases the high-bit into the media, and it can complement the large bit, producing light in proportion to the number of target cells killed. I think one of the most exciting directions of the cell therapy field is moving from autologous products into more allogeneic products. And I think it's exciting because it's an opportunity to standardize the therapies in a way that's going to be beneficial to a lot of patients. It's going to become much more efficient to create large lots of drugs, so hopefully bringing those costs down for patients. And finally, there are a whole lot of people who don't live near, for instance, an academic medical center where they can get an autologous CAR T product. Or their family circumstances, their life circumstances are such that they can't, commit the time and money to that's required. And so I think that these more like off-the-shelf allogeneic products are just going to open up CAR T therapy to way more people, and that's exciting.